I started actually as a medical doctor, and I was really fascinated about the immune system and how it senses the presence of non-self. And this is how I guess I got stuck in immunology. You have to be curious about how things work, inquisitive, explorative, insistent.
Veit Hornung studied medicine before choosing to pursue fundamental research. He now works at the Gene Center of the Ludwig Maximilian University in Munich. His driving force has always been to understand how the immune system works and how it enables living beings to defend themselves against pathogenic organisms. This natural defense is divided into two main systems: innate immunity and adaptive immunity.
Innate immunity you can consider as the primordial, the ancestral immune system that you also find in single-celled organisms. So that is based on receptors sensing the presence of non-self and then triggering a signaling cascade that sets off an alarm in the cell to fight the infections. On the other hand, adaptive immunity is a more elaborate system that you only find in vertebrates that have special receptors that can rearrange in a way that they generate a huge repertoire of different receptors that can sense the presence of non-self structures through T-cell receptors or B-cell receptors. Now, these two systems have been traditionally put as two columns or two entities, but they are actually interacting with one another. The innate immune system instructs the adaptive immune system on how to trigger an immune response, so they're tightly interconnected and work together.
Fundamental research has long favoured adaptive immunity over innate immunity. This defence mechanism has finally aroused the scientific community's interest, with researchers realising its essential key role.
If you don't know which key players play a role there is not much interest. But once the first players, were discovered, and there was also the connection, I think, that the innate immune field is very important. It's not like something we have besides the adaptive immune, right? Which is, the more powerful in the end. But it's actually the priming one. So it's part of the same system. I think when this realization take place, people really knew that this is very important.
Innate immunity is based on the recognition of pathogens by pattern recognition receptors, PRRs. These receptors are able to identify the presence of nucleic acids, that is DNA and RNA from viruses and other pathogens, to signal them and thus trigger a powerful immune response. Veit Hornung's research has provided key insights into how PRRs recognize these foreign nucleic acids.
For me, I find it super fascinating to really look at the cell, like the signaling pathways, and how the cell then facilitates this complex signaling to induce this immune response. And there's so many proteins involved in these signaling cascades, and I think it's important to really understand each one of them.
Right now I'm preparing dilutions of cell culture supernatants. These come from cells that I previously stimulated, and these cells then give an immune response. I just find it super interesting that we have the strongest defense system in our body. It's just sometimes not able to recognize what it is supposed to attack.
Some autoimmune diseases, such as rheumatoid arthritis or type 2 diabetes, may result from a misinterpretation by the innate immune system, leading to harmful inflammation. Veit Hornung's work provides a better understanding of these mechanisms and supports the development of medical treatments for these conditions, and more generally diseases such as cancer.
While our research was primarily motivated to understand how pathogens are sensed, this knowledge can also be translated into therapeutic applications or into understanding how certain diseases work. For example, mRNA vaccines. We can explain why mRNA vaccines trigger immune responses, what receptors are activated, and how we can circumvent this, for example, under certain conditions where we don't want too much inflammation. We can explain how certain autoimmune diseases are triggered, and by identifying what receptors are activated by what nucleic acids, we can specifically target these diseases in the future to come. A very exciting field for us is the use of AI technologies to identify new interaction partners for proteins that we're studying, but also new molecules that might interact with these proteins.
The life science field will probably transverse from a more discovery field into more like engineering type of field almost. With all the AI tools, obviously, right, we can make use of all the data that have been collected much more better. There will be a much bigger transition, I think, from basic discovery into, let's say, more applied life science in the next decade or so.
Computational sciences now support translational medicine, also serving as tools for fundamental research. Today, Veit Hornung and his team are exploring new research methods using artificial intelligence, enabling them to identify cells of interest among millions of human cells.
Once we have identified from these libraries of cells the individual cells that look interesting using these machine learning models, we then, and this is what we do here, go to this so-called laser microdissection microscope, which is basically a laser cutter for cells, and we isolate the nuclei of the cells that look interesting to us.
In the old days, there wouldn't have been any way to sort out these different phenotypes, but with AI approaches, we can use machine learning tools to understand these images, these morphologies, and sort these cells into different categories based on these morphologies. And this is an enormous advance for us. And this is something that is super exciting in the years to come.